Hi guys, I'm Emma, and I'm going to be doing the group video for this week. The group have already discussed all the problems, and we agreed on how to do them, so I'm just going to go and run through my sheet, and we're going to work through all the problems. Okay, let me just go ahead and screen share for you guys. Alright, so here we have our group problems. We're going to start with question 1 and work our way down. So for problem 1, part A, and all of these actually, you just have to do the fork line method. So you take all the A's and you do the Punnett square for them, and that'll tell you the proportions of heterozygous, homozygous, and, well, homozygous dominant and homozygous recessive. And then when you're looking over here at what you want to end up with, all you have to do is combine the different probabilities. So you say, okay, when we did the Punnett square, half of them were AA, so you put that down, and then you go to the next one for the B, and you say, okay, a fourth of ours were BB, so we multiply it by that, and you keep going through, and you're going to do that for each one until you have them multiplied all together, and that's going to give you 1 out of 128, and that will be your proportion. The way you do part B is exactly the same, except with these different combination here, and you end up getting 1 out of 256. For C, it's the same thing again, and you get 1 out of 64. And also for D, it's the same method, you get 1 out of 32. Okay, for part 2, we have a fill-in-the-blank question. So part A says, paired homologous chromosomes are sometimes called what or what? So if you're paired together, that means you have one homologous chromosome and there's the other one. They're paired together like this, and we can call that a bivalent because there's 2 of them. Or a tetrad because you can separate it into 4 parts. For part B, we want to know the structure that connects the centromere to the microtubules of the meiotic spindle. It's called the kinetochore. So the little space that acts like a belt on the chromosomes, making them link together like that, it's called the kinetochore, and the meiotic kinetochore microtubules will come and connect to that. But we already know that they mentioned the meiotic spindle, so they're talking about the kinetochore and not the microtubules. Okay, for part C, it says, a diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell prophase, in prophase 1, has how many sister chromatids, and how many will it have in metaphase 2? So if we take the part with prophase 1, we know we have 4 pairs of chromosomes, and that sister chromatids, if you look at a chromosome, each half is called a sister chromatid. And since we have pairs like this, and we have 4 of them, we're going to take 4 times 2. So there's 2 on each one. We have 4 pairs. 4 times 2 is 8. We should have 8 sister chromatids. And for the second part, where it's in metaphase 2, we have to remember that they have not been separated completely yet. It's still before anaphase 2, so we'll have sets like, here, one sec. So we'll have a cell that has one and a cell that has another. So if we think about it, we should have 4 sister chromatids because we're cutting it in half. For part D, we have to remember what kind of cross you do to determine if a purple flowered plant has a homozygous dominant or a heterozygous genotype. We have to remember that Mendel, when he did this, he took one that he knew, where both were recessive, and he crossed it with his unknown. And he knows that the results resulting offspring will look different depending on what the parent is. And so if you use a test cross, you can determine the genotype of the offspring. Well, the genotype of the parent, I'm sorry. For part E, we want to know what the terms are for a non-functional allele, which we call a null allele. It's null because it doesn't work. It's a dead. And a partially functional allele is called a gain-of-function allele. If you look at all the options listed in the lesson, this is the only one that functions. Ones that can be changed, but they still act the same. So I think that it has to be the one that is different, which would be the gain-of-function allele. Sorry about my voice, I have a cold. For part F, we want to know how to determine which of Mendel's laws involves two traits and which involves a single trait. If we look at the lesson notes, we'll remember that the dihybrid cross was done using independent assortment. How they're found on different chromosomes, and depending on how you arrange it around the metaphase plate, you can get different combinations. So you're dealing with more than one trait. You could have tall, wrinkled, green versus purple, and depending on how it gets split up, you'll know what the traits look like. But we're involving more than one. We're not just looking at height or color. We're looking at all of them at the same time. Whereas Mendel's law of segregation is like what we talked about at the beginning of the lesson with the monohybrid crosses, where you're just wanting to compare if it's tall or short. You're only looking at one trait, but you know that it's going to get one from each parent. It can't get all of them. For part 3, this one is probably the most tricky question for this lesson. So for part A, instead of putting it here in the document, which I would have found kind of difficult, I made a diagram on paper, which I'll show you in just a second, after we look at what we want to find. So it says to diagram all possible homologous chromosome arrangements during metaphase 1 and indicate which alleles are found on each pair of homologous chromosomes. So if you think about it, a homologous chromosome pair, one is from your mom and one is from your dad. And they're going to be lined up on the metaphase plate like this. And because we have 3 different traits, there's going to be 3 different chromosomes. We're assuming they're on a different chromosome for each one. So we'll have a total of 3 above the metaphase plate and 3 below. Then we also need to think about what are our results. What kinds of different pairings of offspring can we have? Which goes to part B. So I did part B first. So you know that each offspring is going to have a big T because it has big T's. And we don't know what the other parent is, and it doesn't really matter since we're just looking at inside them what gametes are going to be formed. So you know all of them are going to have a big T. Then you look and you see that there is a big R and a little r. And so you say, okay, if you have the big T, you can have a big R, and then you can have either a big Y or a little y. So those are 2 options. And then you want to look at the little r's. What's a big T still? So if you have little r's, you can either have a big Y or a little y. So these are all the possible gamete types that we can make. So knowing that, we can make a diagram listing them all. So I took it. I guess I need to stop sharing my screen for a sec. 1 second. So I made this diagram, and each one of these is one of the gamete possibilities of how they can line up. And you have to remember that it hasn't split again, so it hasn't reached anaphase 2 yet. So they're still going to be in homologous pairs. So if you think about it, the ones in yellow, those are your T's. You know that it's going to have a big T no matter what on each side. And so that makes it pretty easy, and you fill all those out. Then you think about your combinations that you had left. So for your R's, you had, there could be a big R or little r, and when you look at the bottom, if you split it in half, that is what is going to be one of your options for the end. You know that it's going to be this combination, so you want to make sure to get all 4 of your different combinations that we just talked about. And then by listing them all out and arranging them in all the possible combinations, you find out there are this many ways to rearrange it, just 4 different ways. Okay. Okay, and part B. Then we look at part C. How many sister chromatids are found per cell in this organism during metaphase 1? So if you look at metaphase 1, we'll notice that there are the chromosomes lined up like this, the homologous pairs, just like we just drew. And for each chromosome, there's going to be 2 chromatids. So let's see, my answer looks like it might be wrong. Okay, so we know we have 6 different, if we look at our picture again, we had them lined up and there were 3 pairs of homologous chromosomes. So we had 6. If you, I'm not doing a very good job. If you look at the arrangement, you have 1 set of chromosomes on the top above the metaphase plate and 1 set below. And you know each of those has 2 chromatids. So if you take it, it would be 2 times 6, and that would be 12 sister chromatids. And then they ask a similar question in part D. They say, how many chromosomes are found per cell in this organism during metaphase 2? So the tricky part about this one is you have to remember that anaphase 2 has not happened yet. So in metaphase 2, they are just, the homologous chromosomes are in different cells, but they're still linked together, so there are 2 sister chromatids per chromosome. So I believe that the same number of chromosomes are found as in metaphase 1, which would be a total of 12. The only other possibility is that it would be 6, but since they haven't split up yet, I still think it's 12. For part 4, we have a horse that has 32 pairs of chromosomes, and we're going through and we're trying to figure out how many chromosomes and such things are there at different steps in meiosis. So for part A, we want to know at the conclusion of meiosis 1, how many chromosomes are present in each cell. If you think about meiosis 1, you take all your chromosomes and it's going to be divided into 2 different parts. So if you're dividing it, you take 32, your starting number, you divide it by 2, and you should have 16 chromosomes in each cell. For part B, how many tetrads are present per horse cell during prophase 1 of meiosis? You remember there are 32 pairs of chromosomes, and each pair is a tetrad, and so you should have 32 tetrads. For part C, we have how many sister chromatids are present in a horse cell during prophase 1 of meiosis? So there are 32 tetrads, and you have to remember that each tetrad consists of the chromosomes with with a total of 4 chromatids. So you have to double the amount of chromosomes. So you have to have 64 sister chromatids, because there's twice as many as there are chromosomes, if that makes sense. Part D says how many sister chromatids are present per horse cell during metaphase 2 of meiosis. So you're thinking about each cell, and again, it's metaphase 2, so they haven't divided yet, so it should still be 32. Now, part E is a little tricky. It's kind of a trick question. It says, how many bivalents are present in a core cell during anaphase 2 of meiosis? Remember that bivalents are when the homologous chromosomes are linked together, so that it forms a tetrad. Because we are doing anaphase 2, they're in separate cells, the different homologous chromosomes, so they're no longer attached in a bivalent at all, so there would be 0 bivalents in each cell. Okay, we come to our last question, which is question 5. And the parents of the offspring and their genotypes, which is very helpful, and then what each allele codes for. So when we look at part A, we want to know the proportion of offspring that's going to be tall, round, green, and terminal. So if we look at this tall portion right here, we know that the tall ones are all going to have a dominant T in them. So the homozygous dominant and then the heterozygous offspring are all going to be tall. So if you do use the fourth line method using a Punnett square, you find that a fourth of all your offspring are going to be homozygous dominant, and half of them are going to be heterozygous dominant for the tall trait. So I multiply those together, and then over here we do the same thing for the round, because we know we need a big R, and so I just separated it to show that there's dominant and the heterozygous. You have to remember to include both. And you go and do the same thing for the rest, and you end up with 1 over 256. If we look at B, we're looking at the genotype. So we're looking at very specific combinations. Again, we look at our fourth line method, and we find that there were homozygous recessive for the T trait a fourth of the time, and they were heterozygous for the R trait half of the time, and half of the time for the Y, and half the time for the A. And you multiply those together and you get 1 over 32. C is the same idea. You just want to know your proportion that are going to be heterozygous for all of them. And all of ours, it's half. So it's half the probability of being heterozygous for all of them, because parents can combine to make heterozygous pairs. So we get 1 out of every 16 is going to be heterozygous for all 4 genes. For D, it's again like part A. You look at the traits they're asking for, and you remember if they're recessive or dominant. You find their frequencies, and you multiply it together, and you get your proportion of 1 over 64. I hope this video was helpful. I'm sorry about the stuttering and the cold. And I hope you enjoy it. Thanks.